Here is a quick introduction and preview to some new features in Curry 9. One of these new features is tractography. If we move to the data set here, we can see a classic DTI image, which is extracted from a special MRI scan sequence. We can combine that with a structural MRI to produce a data set that will allow us to look at changes in fiber tract projection as a function of the location of, in this particular example, an epileptic spike. We'll return to all windows in our display here so we can see the representation of this interictal epileptic spike in the top left, the reconstructed cortical surface in the top right, as well as the DTI fiber tracts and the DTI in the bottom right and the two-dimensional voltage distribution map in the bottom left. One of the unique features of the Curry software is that we can step through that fiber tract projection as an epileptic spike progresses. Now for a moment, let's just come back to our 3D view and we will remove the fiber tracts. In this particular case, we have the example of a propagating spike that begins on the inferior surface of the temporal lobe, but then wraps around the temporal lobe and progresses or propagates on the superior surface. Let's move back to the start of that spike, and then we will reinitiate tractography across that time period. What this provides us with is a view of areas of the brain that potentially could be impacted by the location of the propagated epileptic activity. And we can see how this activity, which starts in the tip of the temporal lobe, could start to explain some of the symptoms that occur as the spike propagates. Again, this is an interictal spike. So as that spike propagates from its location of origin, we could see that many areas of the brain might become involved. This is a simulated example of how we might be able to examine the relationship between the onset of a spike and the propagation characteristics. Thank you.